Hello everybody. Today we're going to be doing a lab to test the action of the enzyme lactase on two different disaccharides. The first one we're going to test it on is a disaccharide called lactose. The second one we're going to test it on is a disaccharide called sucrose. So when you get to your lab station, you're going to notice you have your three solutions. You also have a test tube rack with 4 test tubes, and you have 2 graduated cylinders, a small graduated cylinder and a large one. First thing you need to do is you need to rinse out your test tubes and your graduated cylinders. This is just to make sure that whoever did this lab before you, in case they didn't clean up, you don't want any cross-contamination issues. So I've rinsed my test tubes out already, so I'm going to go ahead and turn these over into my test tube holder. The first sugar we are going to be testing is lactose. Now we're going to be testing the action of the enzyme lactase on these 2 disaccharides by testing for the presence of glucose. So lactose is made up of 2 monosaccharides, glucose and galactose. Sucrose is made up of glucose and fructose. So if lactase is able to break these 2 disaccharides down, glucose will be present and it will show up positive on one of my glucose test strips. So first thing I'm going to do is I'm going to get my big graduated cylinder and I'm going to measure out 10 milliliters of lactose. So I'm going to pour this into my little handy dandy graduated cylinder. Okay, 10 milliliters, and I'm going to pour it in one of my test tubes. Fill this test tube up. Put that back in the holder. I need to repeat this because I need to have a control. Okay, so I'm going to fill it back up to 10 milliliters. I'm going to pour it in my second test tube. So one of these test tubes we're going to put lactase in. The other test tube we're going to not put lactase in. So that one without lactase is going to be our control for this experiment. So I have my cup of lactase. I'm going to swirl it around a little bit. It tends to settle to the bottom. And I'm going to suck some up here with my pipette. And I need 1 milliliter of lactase. So I'm going to go ahead and dump out the extra water. And I'm going to fill this up to 1 milliliter of lactase. Okay, squirt the rest back in my cup. This is going to be my experimental test tube. This one is my control. I pour my lactase in here. I'm going to put my finger on it. Shake it vigorously to make sure it's nice and mixed up, and then I'm going to let it sit. I'm going to set the timer on my phone to let these sit for 3 minutes. At the end of that time, I'm going to get my glucose test strips. Now you will notice when you pull these glucose test strips out that they are actually yellow, not aqua, like the lab packet says. Different glucose test strips are different colors. So I'm going to take a test strip, and I'm going to quickly just dip it in so it touches just my lactose. I'm going to get another one, and I'm going to dip it in to my lactase mixture, and I'm going to let those sit for 3 minutes. That's how long the package says you have to wait before you can read to see if glucose is present. While I'm waiting, I'm going to go ahead and rinse out my graduated cylinders so I know that they are ready for the next part of my experiment. Once you are finished reading your glucose test strips, you're going to repeat the exact same experimental setup with your sucrose to see if lactase does the same thing with sucrose that it does with lactose. Have fun!